Thanks everybody for joining the seminar today. I'm excited to introduce Dr. Tanya Berger-Wolf, who is a professor of computer science engineering, electrical and computer engineering, and evolution, ecology, and organismal biology at The Ohio State University, where she is also the director of the Translational Data Analytics Institute. A pioneer in artificial intelligence for ecology, biodiversity, and conservation, she leads the NSF-funded Imagomics Institute and the US-Canada co-funded AI and Biodiversity Change ABC Global Center. Dr. Berger-Wolf serves as a scientific advisor for many organizations, including the US National Academy's Board on Life Sciences, US National Committee for the International Union of Biological Sciences, the Global Partnership on AI, and the Nature Conservancy. She co-founded the AI for Conservation nonprofit Wild Me, now part of the Conservation X Labs, creator of Wild Book, recognized by UNESCO for advancing the UN Sustainable Development Goals. Her contributions have earned numerous accolades, including recognition as the AI 100 Global Thought Leaders by H2O.ai, and most recently, the OSU College of Engineering Lumley Interdisciplinary Research Award. So again, thanks for joining us, Dr. Tanya Berger-Wolf, and I'm turning it over to you.
Thank you very much for having me here. And I hope this generates discussion. Please do ask questions. And if things really, truly get completely sort of not too dense, not understandable, jargony, feel free to also interrupt me. So with that, let's talk about AI for nature, and particularly this collection of icons at the bottom. I will start with that collection of icons. Most of the work that I will present today is funded and in collaboration and the product of two big projects that I did. One is the AI and Biodiversity Change, ABC, Global Center, co-funded by US National Science Foundation and NSERC Canada. And the goal is to develop AI-enabled tools and analysis to describe impacts of global change on species and habitats and assess how ecosystems respond to intervention. And this is truly a global four-continent collaboration. And the second is Imagomics Institute, funded by the National Science Foundation, with the goal of establishing a new field of science focused on understanding biology of organisms. That's the omics part, particularly traits and observable phenotype, from images. That's the image part. So omics from images, imageomics. So with that, why are we talking about AI for science, and what's the role of AI in science? If we go back to the scientific method summarized by Poincaré in this gorgeous book, Science and Method, he finishes the book with this statement that the scientific method consists in observation and experiment, which is not surprising. This is what we know. And if the scientist had an infinity of time at his disposal, it would be sufficient to say to him, look and look carefully. But since he has not time to look at everything, and above all, to look carefully, and something I tell my students all the time, since it is better not to look at all than to look carelessly, he's forced to make a selection. And so the first question then is how to make the selection. And what I would argue in all of that technology and computing do not fundamentally change the scientific method. What they do is they let us look at more things more carefully. Over the last 20 years or so, this explosion of technology, particularly around our ability to look at more things in the scientific world, is enabled by the satellite development of satellite technology, autonomous vehicles underwater, on the ground, and in the air, on-body sensors that can measure everything from GPS to heartbeats, in situ stationary sensors such as camera traps, acoustic recorders, environmental DNA, and increasingly these devices that we all use every day to take pictures, videos, recordings of everything from what we have for lunch to record the natural world around us. And I bet if this was an interactive talk, I would ask how many of you have taken a picture of a living organism in the last week, reminding that humans are a living organism. Probably everybody. So there's certainly an opportunity to look at many, many, many more things. And in fact, but are we looking actually at all the things that we need to look? And so if we look at the map of the hotspots of biodiversity, which is this top left map, and compare it to the observations of biodiversity in the world. So even through the official data aggregators of biodiversity data, like the Global Biodiversity Information Facility, bottom left, we notice that sort of the hotspots of biodiversity and the intensity of available data are almost complementary in the world. And the pattern is even more pronounced when we move to the citizen science platforms like iNaturalist or sort of general contribution platforms or eBird. The thing is, so the data that we have is hugely biased, not just regionally. So we have most data in the North America and Western Europe with some availability of data depending on the platform, and a little bit in India and a little bit in Western and Eastern Australia. But if we zoom in even more, it's clear that we have more data where in cities and parks and public spaces accessible by roads. And if we zoom even more and look at it from a different perspective, we notice that the highest correlation of data availability on biodiversity is with the average income per geographic unit. So we don't get data from where nature is and the variety of nature and biodiversity. We get data from where rich humans have access to that nature. But anything we put on the x-axis, we're going to have this bias of where we have availability of data, and more importantly, knowledge. So not only we're not looking at everything, we're also not looking carefully at everything. So we recently published this paper in Nature Reviews Biodiversity on the shortfalls of biodiversity knowledge. And not surprisingly, the shortfalls are in every aspect: the stall taxonomic naming and description, the basic sort of description of biodiversity, the abundance and populations estimate, the taxonomic and regional biases of distribution, which we just saw so vividly in the maps, the difficulties of understanding the response of organisms in the environment, morphology and function, the phylogeny, connections to phylogeny, and finally, species interactions. So as we move from just naming to sort of more complex interactions, the shortfalls are more pronounced, the knowledge shortfalls are more pronounced. And we connect them to both the basic ecological field, the subfield, and the conservation sort of impact. But these shortfalls are also an opportunity for AI to sort of to fill that data gap by looking at more data, that knowledge gap, by looking at more data more carefully. And so we outline sort of for each shortfall how it may be connected, how AI may be used to fill that shortfall. And so, but to do that, we can't continue with the standard current sort of approach to designing machine learning and AI methods. And a new paradigm is being advocated for AI in general, for application-driven AI, and which is particularly necessary here in AI to fill the biodiversity shortfalls and the scientific shortfalls here. And what it means is that the AI methods cannot be developed as they are right now, cannot be developed in total absence of the application context. They need, for science particularly, AI has to make discovery possible, and it has to be interpretable. So we need to be able to say, this is the answer because, ultimately generating testable hypotheses. And so with that, oops, we have the current methods, particularly in visual data, which is by far the most abundant and readily available source of information about nature. We have millions and millions and millions of images. We have methods that can take sort of all these millions of images, find all the living organisms, not just animals, but plants as well, although it is very biased towards vertebrates in general. Find images that contain them, identify, localize where they are in those images, and identify not only species, but in many cases down to individual animal for some of them. Great. But what we want is to move from saying that this is a particular species of bird to saying that this is a particular species of bird because has orange belly, black behind the beak, wobbles when it walks, and makes this particular sound and lives in that particular habitat. So that because part is the foundation of AI for science. And so we think that biology is particularly well suited for developing these kinds of methods, and especially images in biologists, because we have an abundance now, by far the most available, readily accessible source of information about especially organisms are images, and not just the RGB sort of images of nature, but a 3D texture model, X-rays, and so on, different modalities coming from different sources, not just kind of the pictures of nature in nature. And they are quite often now increasingly paired with the text description, or we can pair them with text descriptions of species, geospatial information, molecular data, for example, from sources like the DNA barcoding project, BOLD and BioScan. The other part that is the strength of biology is that biology is the science of structures. It is literally the science that kind of leaned in and developed the first taxonomies, but it is also, so that we have the taxonomy of phylogenies. We also have ontologies. There's a whole community that is geeking out on biontologies with the conferences. And so the nice thing is that we have computational paradigms that allow incorporating directly those structures into the architecture of the machine learning models. And that's the knowledge-guided machine learning paradigm. So that the outcome, both it allows us to use less data, actually, quite often, for more impact, less data for more knowledge. And also, it doesn't discard all the centuries of accumulated biological knowledge. And then the outcome of this machine learning also is interpretable, so that the answer is this species because. Right? So let's see. Let's start with the simplest possible type of knowledge to incorporate with images. Taxonomy, right? And so we developed, literally we started, okay, let's take the images that we can and add taxonomy, the full taxonomy, as it is available right now from the taxonomy query engine, as a caption to each image. Does it help? And use the vision language, the multimodal vision language model architecture. And so this is the second version, BioClip, the first foundation model for the tree of life. We took 204, the second version, the first version was released a year ago. This is the second one, 214 million images from Encyclopedia of Life, GBIF, complemented with iNaturalist, then FathomNet, which is the marine data, and BioScan is the DNA barcoding project with a lot of invertebrate images. And so the distribution of different organism groups, images by different organism groups, you see that it compensates quite a bit for the bias towards vertebrates that we had until now. It covers about 1 million taxons, those 214 million images, it's about half of the named species out there. And so it's an unsupervised model, which means we didn't train a classifier by saying, here's an image and it's this. No, we just added a caption of the taxonomy for each image. And so it learns these associations implicitly to any level of the taxonomic hierarchy. Great. So not only it is a great classifier, you can give it an image, not just an image, you can give it a drawing. If it's an accurate drawing, sure. It also can do images of species it has never seen before in training, what's known zero shot, species that are undocumented, non-described new species. It can put them in the typically correct genus or family. But wait, there's more. There are emergent properties. By adding this minimum of biological information, turns out that within the space of the model, where each point is an image, and it is in this multidimensional latent space, embedding space of the model, the model clusters them by species. Great, which is how we can then do inference of a new image, what species it is. But there's also, in different dimension, it separates within species, it separates juvenile and adult, female, male. So it learns without ever being told these emergent properties, something in images actually sort of allows it to group. Within species, it also separates disease from healthy leaves, images of disease from healthy leaves, and separates them by disease. And across species, we said, okay, well, let's see, what happens to Darwin's finches? Well, it turns out that Darwin's finches species are aligned by beak size. The model has no notion of beak size, right? There's no measurement that was ever given, but it is sort of, there is an implicit emergent property that comes out, freshwater, non-freshwater fish, and so on. So it is a good starting point for discovery of a lot. What else is there? There's probably a lot. We've just started. And that's by minimally adding just the taxonomic sequence, right? The information. So it can now be sort of filling gaps in taxonomic, just naming and processing all the information. So colleagues in ABC, David Rolnick at McGill with a group built a camera trap that attracts insects, moths specifically, which is one of the most understudied taxonomic groups. And not only now we can process quickly in known species in the camera trap image, so it's an LED light that attracts the moths, but it also can flag new species, right? And put them in the correct genus or family. Okay, but it doesn't tell us yet this new species of moth because so we've developed whole new architecture, and this is the one technical slide that highlights the parts of the image that sort of support the model's determination for a particular. When you use the model as a classifier, then let's say this image, it says, okay, this is this particular species, why, right? So these are the parts of the image that allow it localize the pixels in the image that were responsible for the model to produce the determination of like Scott Oriole in this case, right? It's the black head, it's the yellow body, and the black tail in this case. So you can use these now to ask what is important for a particular class, or which traits are shared between two, not necessarily just bird, but in general species. What is similar, what is different? And you can play this game, sort of, you can now start using it not just for bird species, but for, domestic animals. It's one of the first ones asked, but even car models. So it's a general method. And yes, moths. So we can now start, this is new species of this is in this genus or this species because here's the part, because it has that, in this case, this white spot with dark localized in the bottom and the top, right? And you can do it at every level of taxonomy. So when you ask, why is it this genus? Well, that's because that's what places it in this genus, right? And so the last piece of the puzzle that allows us to kind of really start looking at more data more carefully, at more things more carefully, is this super quick segmentation and visual prompting. So one of the biggest bottlenecks in using AI today is that it takes so long, especially in images, to annotate every image. If you want to use the traits, right, the anatomic traits, to justify the description or kind of connect to description of a species, then it takes frustratingly long time to kind of segment each part of an organism and label it. And so we developed a method essentially that allows you one-click labeling. It literally is that. So here's an example with a butterfly. You click on that ray, it segments that ray pattern on the wing, and it propagates to the entire, let's say, collection of images, even if they may be different species, looking slightly different. Like in the bottom, here's that white part of the wing. Even if the wing is damaged, it still can kind of semantically propagate. You can do it also to the level of organism. So you can say, okay, I want in all the images, find everything that looks like this, and you provide a mask as a segmented sort of part of the image, so essentially a spider in this case, and it propagates that mask and finds everything that looks sort of like this in the rest of the images. And so and you can see with the birds, it can do pretty, differently looking things, but they're semantically still a bird. And that's because it's not just segmentation, it's segmentation with biological information. Great. And so you can start now processing massive collections to look at things more carefully, right? If AI is good at something, it's at speeding data processing. And so this is the Neon's beetle collection. You can take now a whole drawer like this, segment out each beetle, each organism, separate the background. Here it is, just the organism itself. And then by this one-click thing, you can partition, segment different parts, anatomic parts of the organism. Not just the head, abdomen, thorax, but the whiskers, the legs, and even finer. But for the abdomen, you can also do the same kind of thing to place biological landmarks and propagate them through all the organism, and then automatic measurement of ratios. And we just showed that across the continental scale kind of collection of Neon's collection of beetles, this kind of measurement of morphological traits is more accurate than humans with calipers. And so that allows sort of really super fast processing of collections, hopefully. But the biggest strength of it is not just providing this morphological measurements, but connecting them, the form to function, right? And so we're starting to do this with an event. So it's all of this, everything I presented so far is part of the Imageomics Institute, and we have an event we call Datapalooza, and every year it's focused on slightly different aspects of data. It's a hackathon kind of event. So this past year, it was Funkapalooza, focused on functional traits, where we bring together biologists, computer scientists from all over the world for 3.5 days with the data sets, with the tools, with the knowledge, with expertise, with the questions, all around connecting form to function. And so watch this space. There are several papers coming out of there, several new tools, new discoveries coming out of there, just the event that we had this past August. All right. So we have kind of this whole pipeline from the very, very super fine grain, including we're developing in the process of developing image-based kind of an analog to GWAS, the genome-wide association studies. So this is image-wide association studies connecting phenotype to genotype, and sort of to this very, very large scale models like BioClip. the thing is to change, as I said, going back to change how we do machine learning and AI for science. Computer scientists love benchmarks. So we've developed benchmarks that are not tasks that are machine learning tasks, but tasks that are relevant to biology. So that when a new method is developed for, let's say, new species recognition or new species, flagging new species, then it's relevant to the biological task and measured on the performance on the biological task, not the machine learning benchmark data sets, which essentially don't tell anything new in biology. A lot of the work that we've done comes out of this fantastic and unique course. It's now a year-long course that was started in 2010, and it's experiential introduction to AI and ecology. It was called Field Computational Ecology when we started in 2010. And it's a year-long course. We bring together computer scientists and biologists, ecologists, and then into one sort of setting, give background to both domains to everybody over the course of the fall semester. And then in January, we bring them to the field. For the first 4 iterations of the course, we brought them to Kenya for 3 weeks and said, okay, go and do something amazing. It has to be interdisciplinary. It has to be both kind of ecology and computation and AI, and it has to be sort of field data driven. And it teaches sort of both how is field biology and ecology different from sort of lab versions of it, and what are the questions and the challenges. In 2025, we brought the group to the Hawaii Nian site as the field site in January, PUM. And the projects were driven sort of both by the challenges, the questions, the context, the ecosystem of that location. So that's where the beetle collection processing started, but also the sort of augmenting and looking from a different perspective on the phenology transit and how we both monitor phenology changes and how we connect them, the plant phenology changes, to the bird communities and the changes in the bird and pollinator communities. How we use drones, for example, to monitor the phenology at a sort of regional scale, and much more. And we got lucky. We also saw the eruption while we were there. So talking about drones, so many of the projects that we have came out of these sort of interdisciplinary field-based courses. They found their origin there. One of them is using drones to understand animal behavior, and it became several PhDs. And it allows, so the development of this project allows not just individual sort of one species in one individual animal behavior, but sort of going to the level of population, intra and interspecies interactions. So here's a great example. Not just the displacement of elands by zebras, but there is a displacement and scattering by a giraffe running through. So, you cannot get that level without the ability to look at more things more carefully. And technology not only allows us to literally see the whole 3 species in the context of their environment, but also sort of get to look at it more carefully and extract knowledge where we had gaps and translate them to impact. So we've talked about the basic science until now. And what does AI for science look like? I briefly want to talk about that a lot of what we do, particularly in the area of ecology and biodiversity, the translational aspect of it has urgency, and because of the biodiversity loss crisis, that we're in the middle of what has been termed the 6th mass extinction. We're losing species faster than we can name them. And if AI can help fill that gap, it has to change as well. Wildlabs.net is an online community of tech for conservation. It's a global community bringing together thousands of organizations and individuals who develop or use technology for conservation. Every year, it used to be every 2 years, now every year they do the landscape survey of technology use and technology potential for nature. And every year, including the most recent coming out in the last few years, AI tools are listed as the highest potential, but the lowest, almost the lowest performance. This is the very definition of hype. And so if we do want AI to be useful for nature, AI particularly for implementation, for decision making, so AI for decision support, again, has to connect to domain metrics. I get annoyed when people ask us, so we've developed a method for identifying individual animals from photographs. So the not just zebra, giraffe, whale, or turtle, but Zip is the zebra, Joe the giraffe, and Terry the turtle, and Willie the whale. And people ask, so what's the computer scientist, what's the accuracy of your method? Like, in what? Identifying individual animals from what data? In what context? For what species? From what quality of images? In the wild or in the, a carefully selected one. So all of this is meaningless because I don't care about the accuracy. What I care about how accurately can these methods produce population size estimates? How accurately can it re-identify the same individual across different segments of drone video? How accurately does it allow you to count individual and aerial surveys, and so on and so forth? So how well does it allow me to connect to domain metrics, not to computer vision metrics? And AI that makes the outcomes of AI actionable. Okay, great. So I have identified all the individuals in this image. So what? What are you going to do with this? And are you going to change what you're doing or not? Right? And so we summarized a lot of the kind of the opportunities and the challenges of using AI in biodiversity in the Global Partnership on AI report a couple of years ago now, so GPAY is part of the Organization for Economic Cooperation and Development, OECD. And sort of there are challenges and opportunities for AI and biodiversity across all kind of 4 areas: the data, that we don't have enough data and it's biased and there are gaps, and there are biased gaps, the understanding of the drivers of biodiversity loss, the resources. AI is expensive, and using AI without taking care can take away from the conservation resources that are already constrained. And then the policy and action, and suggesting and evaluating interventions. We cannot talk about AI in this space without mentioning that AI itself, and the question I keep getting asked, is the environmental impact of AI. And I want to very clearly separate that AI is an industry, particularly driven the kind of generative AI, is a huge consumer of resources, especially electricity and water. And it's not just for training anymore, but also for use. So think every time you generate, none of the images here were generated using AI, I'm proud to say. It has a huge CO2 footprint, but 90%, if not more, of what we use in AI for nature, AI for ecology, AI for biodiversity and conservation, is not that AI. It is very lightweight AI. So lumping it together with all of AI is, I think, a little bit of a red herring, but it is absolutely clear that AI as an industry increasingly has a huge environmental impact. And so the outcomes of this biodiversity and AI recommendation interestingly, only one small part of it is that we need more tools and metrics. What we really need is, and number one, is that there is no AI without data. We really need to get our data in order, particularly for global things like ecology and biodiversity and conservation. So we need to get our data house in order before we can even do anything interesting with AI. I can tell you that to get data for BioClip to organize those 214 million images took over a year. And we published the tools for data alignment and cleaning because we had to develop them. How do you deduplicate data? How do you clean out data that's, for different organisms? We found that there's about 10% error in labels, even for kind of highly curated data. So we published all these tools, but that's a lot of effort. And that's, still only tiny part of biodiversity data. So in general, we need to get our data house in order. We need to make sure that we include the communities so that the solutions are developed with, not for, whom they are intended. And there is the funding considerations, of course. And the important part, other than developing tools and metrics, is that the capacity building. So there has to be AI for biodiversity, AI for nature training, and talent development. But the most important question in this space is to ask is it an AI project? Whether it's the basic science and ecology or the impact or the impact and conservation and intervention evaluation. And to unpack it, to really answer that question, these are the four things that we really need to consider. Can you ask the question and make decisions that you need to make already without AI? If not, let's say you have the best possible magic, truly magic AI out there. Will it change what you do? Let's say I can identify every individual beetle in like no matter how blurry images. Will it change? Like to what level we need to push that magic AI? Will it change the science? Will it change your decisions? Will it change the understanding and the actions? And if so, do you have the data? Alarmingly, often I'm hearing now, oh, we can use AI to get the data to because we don't have data, so we can have LLM to give us the data. No, it's the other way around, right? There is no AI without data. So if you don't have the data, you're not going to get, no AI will help you. And then finally, do we have the resources not just to initiate, but to sustain? Because the initial cost of AI is super high. And it's not worth it if you're not going to kind of use it more than once. And so with that, the future of AI, we've built up a variety of tools and variety of sort of methods, but the future of AI is multimodal, multi-scale, multi-sensory. So this is an example of the types of data sets that are starting to emerge. This is the open forest data set from the inventories of individual trees to kind of several trees, regional forests, national forests, and country. So that's the multi-scale. I've mentioned several times that for example, for animal behavior, from individual behavior to sort of pairwise interactions, population level interactions, and interspecies. And hopefully we'll come to ecosystems. And the multisensory aspect of it, we talked a lot about images, but going back to the original, we have everything from acoustic, chemical, even for satellite images and autonomous vehicles. We have the hyperspectral images that give us different sort of aspects. We have sensor data and so on and so forth, and increasingly DNA. So so the question is, how do we put all of this data together into sort of one understanding, into one setting? And so we are building an experimental smart lab. We're starting with our own sort of almost backyard, the wilds, which is the largest safari in North America, 10,000 plus acres, but it is about an hour east of Columbus, Ohio, where I am. And it does have zebras and giraffes, native ecosystem. It also has beetles and salamanders and a lot of native species as well. It's one of the biggest conservation science facilities. And so we're instrumenting it with different sensors, different modalities, so that we can start building the multi-model models that take advantage of our ability to kind of see more things, look at more things more carefully, everything everywhere all at once, while minimizing the impact of the ecosystems. Because we need to, on the technical side, we need to test, benchmark, experiment, calibrate, model, and visualize data so that we are able to truly do the science that we want to do, to explore, to study, to understand, discover, predict, and engage everybody in science. So the wilds, we've done a pilot study this summer, particularly the entire month of August, and it kind of builds on what we're continuing to do at the Hawaii Neon site, where the phenology transect, we have camera traps pointing at each plant there and paired up with acoustic sensors. And we did fly drones over that, but the two modalities plus the human sort of transect are continuing. So we'll kind of start building these multimodal models. For the smart wilds, we have 80 GPS tags on paired Davis deer, functionally extinct species, with camera traps, drones, and acoustic sensors. And so here's an example of the camera trap and a drone view of the population at the same time. You can see that drones definitely allow us to see a lot more things at the same time. And we're starting to build a digital twin, so that will allow us to kind of not just visualize the data, but play the what if scenario to train the models before we take them out into the actual environment. So this is an example of the view from also, again, these are takes a couple of hours to build. So these are horse behavior, not zebras. They're dressed as zebras, but sort of a camera trap view versus a drone of the same. And so we can now start placing the synthetic camera traps in different location and telling us, okay, what will the camera trap data tell us about the population interactions if we place them here versus there? And allow us to move ultimately, if we really want to use AI for both science and understanding the ecosystem response to the global and local changes, we need to move AI from sort of primary data collection, to get more data and process it and scale up and speed up data processing, to really focusing not on objects like individual organism or individual or collections of organisms, but interactions and system level understanding. And that's where AI truly is maturing to be able to try, so to be useful. It goes hand in hand with developing new AI technology, AI methods, AI approaches. So that's AI research that is in context of science and decision support. And so with that, the work that I presented is a work by many collaborators, particularly from my own lab, students from my own lab, the Imagenomics Institute, AI and ABC, AI and Biodiversity Global Center, and the Wild Me, Conservation X Labs, and many volunteers around the world and collaborators and partners who ultimately actually do the work in the field that allows to build these digital methods and understanding to bring them back to the field. Thank you. I'm happy to answer questions. Okay.
Great. Thank you so much, Tanya. I was looking for the virtual applause hand, but don't see it in my little bar down
Yeah,
here.
there's thundering in my
All right, so we
um,
have a few questions in the question and answer window. And again, I'll remind folks, feel free to either ask questions in the question and answer button at the bottom menu in Zoom, there's the Q&A button, or you can raise your hand.
yeah, so I can start answering the questions that are already maybe in the Q&A. So the first question about how accurate do the initial taxonomic captions need to be? I assume that's about BioClip 2 or the whole sort of family of BioClip. So as I mentioned, about 10% of captions were incorrect. And of course it matters, and it didn't throw off the model. Some of them were not even resolved to the level of species. Some were to the level of genus or tribe, or depending on the taxonomic group. And if we have sufficiently many examples, of course, then a few errors don't matter. But if this is the only image that we have of a particular species and it is mislabeled, it's going to matter a lot. But also, what do we mean by accurate? Right? The model was trained in a particular state of taxonomy, which is probably going to change tomorrow if we were training it again. One of the biggest shocks for computer scientists was that taxonomy is in flux, that it's not fixed, and it's also not the same depending on who you ask. And so we document very well what is the state of which taxonomy at what point and with what kind of query we're using to get the labels. And ultimately, nothing is 100%. So do the errors propagate? To the same extent that the sort of current taxonomic definitions propagate to the rest of our ecological and evolutionary knowledge and research that's being done. And if it changes tomorrow, we'll have to retrain the models. The question is it useful BioClip 2 and for what inference, right? So I worry less about the accuracy and the results. So the results, it's a foundation model. So you can use it, for example, we're using it as a starting point for tick identification. We don't have a lot of tick species currently in BioClip, but it is a starting point where you fine tune when you augment it with other data for a lot less resources, right? So it's much cheaper than to train a good classifier starting with this. It's been used as a starting point. BioClip version 1 already was used as a starting point for tree species classification from canopy images, and so on and so forth. So it's a starting point. The current model, the current version 2, is also a starting point for exploration. So the result is a hypothesis that you still need to test, right? It's a potential pattern, and that's where I worry a lot less about how accurate it is, but does it tell us something new that we can then test with experiment? Okay. So the question is, interested in gathering secondary data from nature images. iNaturalist, can vision transformers prompt can be employed to acquire secondary data beyond species identification and traits? For example, can the model be prompted to describe or capture species interactions? So we're going that way. And actually, there is a great work by Sarah Beery from MIT doing exactly that and going in exactly that direction. Was of my hidden slides. Let me find it and unhide it and just kind of maybe at least flash it. It's the work called Inquire. So this is Sarah Berry, MIT, and that's Inquire, and that's going exactly at iNaturalist images and finding sort of the interactions, such as a hermit crab using vision language models to provide language queries to find these kinds of interactions, right? So, and exactly things like, oh, a particular type of maybe bee species pollinating a particular flower, or in general, find all the solitary bees pollinating flowers or something like that. So that's exactly the work, the direction that they're going. And I encourage you to take a look there. Can you elaborate on how you see the AI tools support conservation efforts, both locally or even globally? Yeah. So I have a whole other big talk that's focused explicitly on conservation, and maybe that's a different. So we really focus on several aspects. Biodiversity monitoring is the biggest effort, and in general, monitoring is the kind of lowest hanging fruit. The probably most used AI tool in conservation today is, again, Sarah Beery's mega detector, and that's a super light model that simply allows to process camera trap images and find the ones that have something in them, like an actual animal. Used millions of times and millions of images monthly right now. There's a growing number of organizations that take research tools and translate them into conservation kind of research level methods in AI and translate them into conservation effort. We've started with WildMe. So WildBook is a platform that used our algorithm for identifying individual animals from photographs, and then enabled a whole platform that is used now for monitoring population sizes, translating literally providing population size estimates and tracking individuals from whale sharks and manta rays to weedy leafy sea dragons and Hawaiian snails to zebras and giraffes and big charismatic species and leopards and jaguars. It was used for the first census of gravy zebras, the endangered zebras, by just photographs of people driving around the country for 2 days. So, but what want to say is that there is an increasing number of these organizations and organizations working together. The best example of the use of technology, and increasingly AI for conservation, is really the Wild Labs community. And so I see what is really, to me, kind of exciting to see is that we are moving from the kind of basic monitoring, basic processing of data using AI to decision support, to finding where's the best investment, what is the best places to connect the habitats, by putting together the entire stack from satellite images to the super fine grain kind of monitoring data.
Thank you so much. I think we are going over time. I know, like you said, that could be a whole nother talk, but that was really, really fascinating and thought-provoking. Tanya, thank you so much for sharing your work and your perspectives with us. What a wonderful talk. Thank you everyone for attending. We do have another seminar coming up in November. I put the link in the chat, so we'd love to see you there. Thanks again for a great talk and enjoy the rest of your day. Thanks everyone.
Thank you.